First of all, thank you. I want to thank Professor Lederman and Professor Margolis who arranged this visit. And today's talk, actually, the Professor Lederman is one of the, I think, leader scientists in the immunology of HIV. Thank you again. And I hope that students will be really excited about this talk, and very short, maybe, introduction by Professor Margolis.
Hello. I must mention that today is might be a milestone in the programs of our faculty, because it's not just Professor Lederman lecture, but we start of international series of lecture, right? International, where most prominent, we will invite most prominent, yeah, Dr. Ronald. I say that we start a series, international series of lectures in molecular biology, immunology, and virology. And we will invite once a month, once in 2 months, most prominent virologists, immunologists, molecular biologists from abroad. And I have already a list of people who want to come to the next one, and also after the next, from the United States, from Italy, from France, from Czech Republic, and maybe from other countries. So today is the first lecture in this series, and I have an honor to introduce Professor Lederman. Professor Lederman is immunologist and virologist, and definitely one of the leaders in this community in the United States. As you see, he has many affiliations, and if I would read his CV, probably will finish in 2 hours, because it's full of his position, honors, and etc. I must say that also Dr. Lederman is a leader of some community to which I'm proud to belong, a community of virologists which come together in Cleveland, the city where he works, and talk about the most recent accomplishment in this field. So I wouldn't spend more time. Also, I must mention that Professor Lederman is the editor-in-chief of a very good journal. He may talk about it himself. But anyway, I'm proud to belong to this community, which Professor Lederman is a leader. Yes.
So, thank you, Dr. Margolis, for that kind initiation of this series of lectures here. And thank you, Professor Ilijeva, for inviting me here. I've been studying HIV for my entire career. And so, for me, it's a good model to talk about the pathogenesis of a pandemic viral infection. So, why does a virus make you sick? It might be in the interest of the virus to make you sick if the sickness offers an opportunity of dissemination to other people, of replication in general in the host, in agility, that is the ability to evade host immune defenses. And it's kind of a strategic thinking that a virus must engage in, because there must be a balance between tolerance of replication errors, the mistakes that a virus makes in order to evolve, and evolutionary adaptation. And HIV is very good at this. So, one of the things that characterizes HIV infection is that the immune system is profoundly activated, and it's inflamed. And you can see how inflammation can promote acquisition of infection, and I'll tell you why that is in just a moment. And is that in the virus's interest to do this? And the answer is, maybe it is, but the fact of the matter is, it's not necessary. HIV preferentially infects activated CD4 positive T lymphocytes. And it replicates more effectively in activated CD4 positive lymphocytes. So you could see how it might be in the virus's advantage to promote inflammation and activation. But it doesn't seem to be necessary for all related lentiviruses, because HIV is derived from SIV, which is a virus that is prevalent in African non-human primates. And amazingly, those primates have largely managed to tolerate SIV without much of the illness that characterizes HIV, without much of the inflammation and activation that characterizes HIV. But the issue here is that humans, which are an accidental host of SIV becoming HIV, in humans, inflammation and activation are linked to morbidity and mortality. So I'm going to shift gears just a little bit and ask you a question. How do complex organisms like us survive in a world that's populated by rapidly evolving pathogens, while our ability to evolve is very, very, very slow? How do we manage? How do we survive? If you're going to evolve in response to a selection pressure, you need to do a couple of things. First, you need to reproduce your genome, obviously. But you need to make mistakes in copying that genome. And that implies that the mistakes that confer a survival advantage are the ones that will evolve and survive in the presence of any selection pressure. So we're not very good at this. It takes us about 12 years or more before we're even capable of replication. Bacteria like E. coli can take about 20 minutes to half an hour to double. And a virus like HIV, in a single replication cycle that can take hours to a few days, can produce tens to hundreds of thousands of viral particles that can then go on to infect other cells. What's more, the enzymes that catalyze our reproduction, our DNA polymerases, are very faithful. They don't like to make mistakes because we kind of like it the way we are, and we want our children to pretty much be the way we are. And so they make mistakes once in every 10 to the 8th or 1/10 to the 10th reads. In contrast, HIV that has a genome of only 10,000 bases can make on average one mistake with every replication, 10 to the minus 4. So the question is, how do we, humans, manage to deal with this amazing evolutionary adaptation disadvantage that we have? Well, we've got an immune system, and I asked my daughter when she was 8 years old, I asked her this question, and she looked at me and she said, well, we survive with our wits. Well, maybe we do, but in fact, it's our immune system that antedated our wits. And the immune system can be divided in a simplistic way into innate defenses that are dressed and ready. And what do I mean by that? Innate defenses have the brilliant development of what's called pattern recognition. They've divided the world of microbes that have it in for us, that don't like us, into a dozen or so different patterns. And each microbe, virus, bacterium, fungus, whatever it is, has within it or on it some pattern that is recognized by the innate immune system that activates those cells. And the beauty of this is that these innate receptors are dispersed throughout the body. There's lots and lots and lots of them. But then we have this adaptive defenses, which is what we call the adaptive immune system. And it's a way of learning, and it's a way of evolving, but it does so in a different way. It does so via having the characteristics of memory and specificity. What do I mean by that? If you've seen one structure for antibodies, peptide sequence for T cells, if you've seen it, and it's immunogenic, and most of them are, many of them are, then you remember it. And if you see it again, you have a burst of replication of cells that either make antibody or mediate host cell-mediated defenses. And there's also specificity, which means that typically an antibody or a T cell receptor that recognizes peptides in the context of HLA is very specific for that sequence or for that structure. Now, there are exceptions. There are cross-reactivities occasionally, but largely they are specific. These defenses target both extracellular organisms, largely bacteria, and intracellular pathogens, T cells. And how do they achieve diversity? Well, they do it in a couple of ways. One is by rearrangement of their genetic information inside maturing B cells in the bone marrow. And they do so in the thymus for T cell receptors. And in some settings, specifically for antibodies, in the lymph nodes, you can have evolution in real time, such that a B cell receptor, an immunoglobulin on the surface of the cell, will recognize a structure. Many B cells are likely to recognize the related structure of an organism. And then as that B cell matures and moves into the follicle, B cells with the greatest affinity for that structure will outproliferate those with lower affinity, and then we have then we result in the development of a repertoire of antibodies made by these B cells with the highest possible affinity for those structures. And that's what I mean by affinity maturation. And finally, we have plasticity. What do I mean by plasticity? If you're going to fight 1 billion microbes that are giving you trouble for whatever the reason, you need to have enough soldiers to do the job. But if the adaptive immune system, which I've already told you is very specific, if it's very specific and can encompass more than billions of different potential responses, if we were always filled with enough soldiers to do the job, we wouldn't have enough room for other organs. We wouldn't have brains, we wouldn't have hearts. You've got to be able to expand when you need it and contract when you need it. And that's what I mean by plasticity. And when we talk about the ability to expand, we're talking about cell memory, and that is animation. Now, you don't need to know everything on the slide, but the point is that the host defenses that we have reflect complex interactions among the immune cells. They're messengers, typically what we call cytokines, molecules that travel from one cell to another. And their job is to protect the other human cells that they have sworn on their lives to protect. When that system fails tremendously, as it does in the context of HIV, we get infections that we call opportunistic. By opportunistic, we mean that these are typically infections that don't bother the normal host, but in a major immune compromise, they can produce disseminated infection, such as you can see on the right with these acid-fast bacteria. Just billions of them, billions and billions of them in the organs of the host. So this was the first published report in a peer-reviewed journal of the AIDS pandemic. It is now 43 years ago, nearly 43 years ago, where pneumocystis carinii, now Juravecki infection, mucosal candidiasis, is observed in previously healthy hosts. This was unusual. People who developed these infections typically had immunocompromise mediated by chemotherapy or had heritable, recognizable, congenital disorders of the immune system in previously healthy adults. Previously healthy adults with no other explanation didn't get these infections. And I want to point out that one of the key characteristics of HIV was a decrease in the numbers of circulating CD4 positive lymphocytes. And what's more, another characteristic of in these first observations was that there was enhanced expression of a cell surface antigen that reacted with an antibody called OKT10, and now we call that CD38, which is an activation marker. This is one of my mentors, Oscar Radnoff, who burst into my office in July of 1982, telling me, and he was a clotter, he studied coagulation, Hagerty factor was discovered by him, And he came into my office and threw down this Morbidity and Mortality Weekly report and said, look, there's AIDS in hemophiliacs, and you've got to do something about it. I was doing something else at the time, and I was following the advice of my mentors, and I was focusing on my work, which was the pathogenesis of type 1 diabetes. But he was irresistible. He was irresistible. He persuaded me to do some studies to see why hemophiliacs might have acquired this AIDS-like syndrome. And what we found, and published later, actually in January of 1983, was hemophiliacs who were treated with anti-hemophilic factor concentrates that was prepared from the plasma of thousands of donors around the country was linked to an AIDS-like immune impairment. And I won't go over the specific phenotype and functional abnormalities, but they were pretty much the same as we saw in people with AIDS. And these were asymptomatic people. These were hemophiliacs who'd been treated with these pooled concentrates. In contrast, hemophiliacs treated with single donor cryoprecipitates had normal immune function. So if you got maybe infusions of clotting factor prepared from a dozen or so different volunteer donors, you were likely to not have these abnormalities. But if you got pooled factor prepared from thousands and thousands of donors, you would. And so we concluded that the AIDS agent, whatever it was, because it had not yet been identified, was transmissible through pooled plasma products. A bit later, Barbara Kroner at the National Cancer Institute showed that treatment intensity determines the risk for HIV infection in hemophilia. So those who had no treatment rarely got infected. These are proportion HIV negative. And those who were heavily treated or moderately treated, virtually all acquired HIV infection by 1983-84, early on in the epidemic. So how does HIV infect cells? Well, up high, we have the virus, and down here, we have the cell membrane. The first thing that happens is that the envelope glycoprotein binds to CD4. It undergoes a conformational change, which allows binding of the envelope to a chemokine co-receptor, typically either CCR5 or CXCR4. And then that opens up, and the GP41 inserts itself into the cell membrane and fuses with the cell membrane, allowing the dispersal of viral core elements into the cell. So we wanted to see what it was that protected the few 5% of hemophiliacs who had escaped infection up until 1983, and we call them high-risk seronegatives, HRSN. And amazingly, 16% of them, 7 out of 29, were homozygous for a 32 base pair deletion in the CCR5 open-read frame. So I think what others, Paxton concluded, what we concluded, that some humans have dealt with HIV by intrinsic resistance. And about 1% of persons of European origin are homozygous for this mutation and are almost completely resistant to HIV infection. And I don't think that was our intent. That's not what we were thinking of. But what I don't understand is why the allele frequency, which is up to 15%, 20% in some places, are heterozygous for this allele. And I don't understand why it's so prevalent in Europeans. And I don't think anyone has explained this yet. People used to think that it provided resistance to plague, but those data could not be reproduced. So what's the importance of HIV co-receptor CCR5? If it's absent, it protects against infection. We can blockade CCR5 with ligand analogs, which we did with an agent called PSC-Rantes, and we protected all macaques who were treated with high concentrations against SHIV infection. And we can induce allosteric blockade of CCR5 with a small molecule called baramarok that can be part of an HIV treatment regimen. And interestingly, and I'm not going to talk too much about this, if we replace the immune system of a person with HIV with donor cells, stem cells, taken from a CCR5 knockout individual, we can cure HIV infection. This has been done about half a dozen times. much more palatable, much simpler, with lower morbidity, is we found that if we treat with combinations of antiretroviral drugs, we can induce sustained suppression of HIV replication. And this is just a graph of the viral load in plasma and the decay of plasma as a consequence of antiretroviral therapy. But, and I hope you don't mind if I just move over to sit a little bit, because as you saw, I'm emeritus, and emeritus means old. So I'm going to sit down. We can pretty much eliminate HIV from plasma with combination antiretroviral drugs, but HIV also integrates its genome into the genome of resting memory CD4 T cells. And this reservoir, this latent reservoir, persists for years and years on what we're calling HAART, highly active antiretroviral therapy. It's a brilliant concept. It's a brilliant thing that the virus does. These latent sequences, if they're not expressing viral RNAs and proteins, are impervious to host defenses. Once integrated in that cell and not transcribing or translating into proteins, that cell, which contains potentially infectious virus, is impervious to host defenses. But if there's ongoing low-level replication, the high error frequency of the HIV reverse transcriptase allows evolution in response to selection pressure, even antiretroviral drugs. So drug resistance, when given in an incompletely suppressive regimen, is very common. So what this means is that the reservoir itself is a backup that can periodically be updated if necessary. So back to the original question. How does HIV make you sick? It's pretty clear that ongoing viral replication drives progressive immune deficiency that's characterized by a loss of circulating CD4 positive T cells. And we know this by work by Brigitte Tom in France and my lab in the States, that if we suppress HIV replication with combinations of antiretroviral therapies, the immune system recovers, but not always completely. So how does HIV actually make you sick? In earlier studies done in the multi-center AIDS cohort study, we could see that with the y-axis being the CD4 T cell decline, so more rapid decline, and viral levels in plasma going this way, that the higher your virus level in plasma, the more rapid your CD4 decline. So this was first shown by John Mellors in the MAX cohort, and then following that in our cohort, which is the study population I'm reporting now by Benigno Rodriguez, that these lines were virtually superimposable. But here's the twist. The correlation between CD4 T cell decline and plasma RNA, when you look at individual data sets, is weak. The R squared is only 0.09, which means that only 10% of the variability in the rate of CD4 decline can be attributed to the magnitude of viremia. So we're left with this issue: how HIV infection and replication drive progressive immune deficiency, at least in part through intermediate mechanisms. So what are these mechanisms? This is the group that Dr. Margolis is a part of. This is a group called the Bad Boys of Cleveland. You can see us at our leisure at a meeting. And we're called the Bad Boys, and all these characters up here turn out to be men. But we have girls too, and we just didn't show them in this picture. This is before all the stuff that people are talking about nowadays. But anyway, so we have boys and girls in this group, both. And you can see Dr. Margolis is right here. He's looking to his right. I don't know why he's looking that way. He's not looking at the camera. Maybe he's a little shy. So one of the things that happens early on in HIV infection is that the gut is rapidly depleted of CD4 positive lymphocytes. So you can see these stained brown cells are CD4 positive cells in an HIV negative person. And here we have a gut biopsy from an HIV positive person. And this work was done by Jason Brenchley and Danny Dueck's lab, part of the BBC. And similar findings were found by multiple other groups, several other groups, actually initially in the SIB model by Ron Veazey. But importantly, this lesion is rarely corrected with antiretroviral therapy. So even though we can bump CD4 positive T cells in circulation, the gut CD4 depletion tends to persist. And what's the consequence of this? Well, the consequence of this is that the ordinary mucosal barrier to translocation of microbial products is impaired. So levels of LPS, lipopolysaccharide, a component of the cell wall of bacteria, is expanded in plasma. Higher levels in plasma in people with chronic HIV infection and in people with AIDS. And importantly, the CD4 T cell restoration that we see on antiretroviral therapy is inversely proportional to the levels of bacterial products. So on the left, we have the levels of bacterial DNA in plasma. On the right, we have the levels of LPS in plasma. Now the axes are changed, but the point is for each of these high levels of bacterial products in plasma correlate with weaker CD4 T cell restoration on antiretroviral therapy. So it turns out that even though we can suppress viral replication, there are complications of chronic infection that are associated with persistent immune defects and are linked to morbid outcomes. And here's a study that we did in the AIDS clinical trials group showing that multiple plasma markers, interleukin-6, IP10, interferon product, tumor necrosis factor receptors, soluble CD14, coagulation products, D-dimer, are all linked to an increased risk of morbid outcomes in both malignancy, cardiovascular disease, and death. And each of these relationships is significant. So what we end up with is that there's a generalized pro-inflammatory soup in antiretroviral treated persons with HIV infection. Now, what's responsible for this soup? Well, there are many factors. One, on the bottom, we have microbial translocation that I mentioned. Two is the presence of co-pathogens like cytomegalovirus, which is prevalent among 90% of gay men with HIV. We have perturbations of fat metabolism in HIV. We have residual HIV production, which we can demonstrate even in people on antiviral therapy. We have homeostatic responses to lymphopenia that contribute to an inflammatory environment. And we also lose regulatory T cells, whose job it is to regulate heightened immune activation. What we find in people who fail to restore their CD4 T cells, those are the characters in red, is high levels of these inflammatory mediators, IL-6, lipopolysaccharide endotoxins, soluble CD14, which is a surface marker of monocyte macrophages, and the coagulation factor D-dimer. What's more, CD4 and cycling is increased. That is, and we can measure this by expression of the intracellular antigen Ki-67 in persons with immune failure. And importantly, this CD4 cycling and death, and I'm not going to show you the data, can be driven by interleukin-6 and by interleukin-1. And here's a way that we can measure cycling ex vivo. We label cells that are CD71 positive. CD71 is a marker that correlates very well with Ki-67, so this allows us to put in a dye that when we measure on a flow cytometer, movement to the left reflects proliferation, dilution of dye in the cell, hence proliferation, hence intensity is diminished. And we see that proliferation is seen in the CD71 positive cells, not in the CD71 negative cells. And people with healthy controls and immune responders to antiviral therapy have good proliferation. But when we look at the immune non-responders, people who fail to increase their CD4 T cells, there's a failure of proliferation and those cells die. So HIV infection, both untreated and treated, is an inflammatory disease, and that is linked to morbid outcomes. So how do we know which of these multiple inflammatory mediators are on the causal pathway of disease and which are bystanders? And this is important if we want to prevent some of these morbid outcomes. If we target these inflammatory pathways and we target the right pathways, we can preserve health and maybe even cure HIV infection. So we did a study a couple of years ago targeting interleukin-6 using an antibody to the interleukin-6 receptor, tocilizumab. And why did we choose IL-6? First of all, when we looked at all the multiple factors that predict morbid outcome and death in HIV, plasma levels of IL-6 were the most robust. We have shown that cycling of CD4 T cells is dysregulated in vivo, particularly in immune non-responders. I showed you some of the data from the Immunes paper. And IL-6 drives CD4 positive T cells into dysregulated cycling, makes them want to go into cycle, but they fail and die. Interleukin-6 also decreases the expression of the interleukin-7 receptor and interleukin-7 responsiveness. And why is this important? It's important because interleukin-7 drives CD4 T cell homeostasis, promoting cell survival and trafficking. So this is this crossover trial that we did. It was a placebo-controlled trial with arm A initially getting, and it's a double-blind trial. Arm A gets tocilizumab, 3 doses injected. The placebo group gets a placebo injection. And then at week 10, we have a washout period, and there's crossover. So we can see the effects of tocilizumab in each group. This allowed us to have more power to sort out differences by doing it twice, separately in each group. Our primary objectives were to look at the safety of blockade of the receptor in persons with HIV, that's PWH, look at the effect of the intervention on C-reactive protein levels, a classic marker of inflammation and activation, and the effects of the intervention on dysregulated T cell cycling. And what we found is that in the blue shaded period is the period in which people got tocilizumab, arm A, arm B, and during tocilizumab administration, CRP levels fell dramatically. But importantly, and that was our primary objective, but in addition, all these other markers that are linked to morbid outcomes in HIV disease were decreased by tocilizumab. That's soluble CD14, soluble CD40 ligand, D-dimer, tumor necrosis factor receptors 1 and 2, and interleukin-7. And that's an interesting one that I'll get to in a moment. So, to summarize the findings of this study, when we blocked interleukin-6 receptor by tocilizumab in people who were already on antiretroviral therapy, we showed profound decreases in circulating inflammatory indices that are linked to morbidity and mortality, correction of dysregulated T cell cycling, I didn't show you those data, and decreases in plasma levels of the interleukin-7, probably by upregulation of the IL-7 receptor alpha chain CD127 that allows, to which plasma IL-7 will bind and get internalized. That's our interpretation of the data, and that was just published last year. So, why does HIV make you sick? I think the reason that HIV makes you sick is that we, humans, have not yet learned to live peaceably with HIV infection the same way that non-human primates do with SIV. I think it's fair to say that HIV infection, both treated and untreated, is an inflammatory disease that targets immune cells. And I'm accusing interleukin-6 of being an important driver of the pro-inflammatory, pro-coagulant environment of chronic HIV infection. And even though interleukin-6 is a vital cytokine for immune homeostasis, it may not serve our interests in persons with HIV and shorten our survival. It also may not serve the virus's interests entirely by blocking IL-7-mediated CD4 T cell homeostasis and persistence of the reservoir. So these are the people who've done the work at Case Western Reserve, people in my lab, Scott Sig, Mike Freeman, Jeff Jacobson, Len Calabrese, my collaborator at Cleveland Clinic, other folks at CWRU, specifically Janelle Salkowitz, Benigno Rodriguez, Carrie Scheid, who were in my lab. And the BBC has broad representation. Dr. Margolis is one of our charter members, Nick Funderburg, Alan Landay, Mirko Paradine, and others. This work was funded by the AIDS Clinical Trials Group at the National Institutes of Health, the Fazemeyer Foundation, and the Scott Inckley Endowment, and the Feldstein Endowment. I want to talk to you a little bit about this journal that I am the editor-in-chief of. It's called Pathogens and Immunity. And in my mind, there's no other journal in the world like it. First of all, it's free and open access. So anybody can get on the website. It's pajournal.com. No charge. Everything on all our contents are free. What's more, in addition to the free open access, there are no fees to authors. So if you submit a paper to us, it doesn't cost you a dime. Submission online takes less than 5 minutes. How many of you have spent more than 3 hours trying to submit a paper to another journal? We also pay our reviewers. So the people who review for us get a modest honorarium. It's not a lot of money, but they will be paid if they can get a review to us within 10 days. As a result, our editorial decisions are rapid. The median time from submission to an editorial decision is 14 days. We're fully indexed on PubMed, and we have a citation score of nearly 12, and we're ranked in the top octile of more than 300 infectious disease journals by Simago. And our citation frequency in the past 2 years was higher than the Journal of Immunology, higher than the Journal of Infectious Diseases, higher than Open Forum of Infectious Diseases, Antibiotics, AAC, and JAC. And when we launched, John Cohen gave us a really great review talking about what an interesting journal this is. So I want to thank you for your attention, and if you have any questions or you want to throw fruit at me, that's fine too.
Thank you very much for a very interesting presentation. I'm not in this field, but I would like to ask you, what is the future of humans and HIV? Can we eliminate this virus at all, or we will adapt, or what would be your thoughts about that?
That's a great question. And my response to you is, how much time do you have? How long do you want to wait?
Yeah.
If you want to wait for adaptation to HIV, my guess is it's going to be too long. If you want to wait for our wits, as my 8-year-old daughter suggested, to conquer HIV, well, we've gotten partway there. With antiretroviral therapy, 1 pill a day, people can have a normal or near normal life expectancy if they take their medications. Now, making a vaccine for HIV is a little more difficult, and we haven't quite gotten there yet. And there are a lot of reasons for this, but we can make good monoclonal antibodies that can neutralize HIV. But the problem is that from all these antibodies, there are viruses that are not susceptible to them. Some are broadly reactive that target multiple different kinds of HIV variants. And the question is, can we induce those responses by immunizing humans? That gets to be very complicated, because earlier I mentioned what happens to B cells as they mature, this concept of affinity maturation in the lymph node. It turns out that those humans who have broadly neutralizing antibodies against HIV, those antibodies have undergone profound affinity maturation. So it's not easy to sculpt those antibodies. It's not easy to do it. immunization regimens look to be a bit complicated. It's not that they're beyond the realm of possibility, but they're going to be a little difficult. But I have to say that the strategy that strikes me as most effective in terms of preventing HIV with neutralizing antibodies is putting those antibodies into a vector that can get into human cells on an episome, for example, that is replicating, that results in sustained expression of antibodies by our own factory cells, muscle cells. I think this is plausible. This is likely to happen. Now, the third one is, can we cure HIV? And that's going to be really tough. I think that curing HIV is going to be very, very difficult. Even though there have been a handful of people who have been cured with CCR5 knockout stem cells, I think that the complexity of that approach, 20% mortality, is very risky. And all things considered, it's easier to take one pill a day. Or an injectable antiretroviral.
The ratio of CD4 plus and CD8 plus are changed during the developing infection and during the treatment also. My question is how the interleukin-6 effects on the change in this ratio.
So I don't think interleukin-6 is a critical determinant of the CD4-CD8 ratio, other than driving CD4 T cells into cycle and death. So in that way, it can contribute to a low CD4-CD8 ratio. So it can do so via an effect primarily on CD4 cycle and death. But the CD8 expansion that you mentioned, and it is an absolute expansion in early infection, is a consequence of several things. One is cytomegalovirus infection, that CMV is an amazing virus. Even in healthy HIV uninfected people, it promotes CD8 T cell expansion. And the other thing that promotes CD8 T cell expansion, and both of these arenas are things that my lab has worked on, is expression of interleukin-15. So high levels of interleukin-15 in certain sites, particularly vascular sites, drive CD8 expansion. Cytomegalovirus drives CD8 expansion. And interleukin-6, as you suggested, can drive CD4 T cell decreases. But the loss of CD4 T cells is multifactorial. It's not just IL-6, it's IL-1, and it's also the effect of the virus itself is responsible for the death of some CD4 T cells.
Thank you so much for your work and for your presentation. So I have one question about some medicines and cure, because yesterday's Nobel Prize ceremony, we are influenced with that, and my question is about microRNAs and your view about that. If you see any kind of perspectives regarding microRNAs and HIV.
I'm embarrassed to say that I can't say anything about that. I just don't know. So can you tell me what you think might be happening, would be a potential application of microRNAs to HIV cure and therapy? What do you think?
Since morning I was thinking about that, really. I was really influenced because of that, and I think that we need some research regarding HIV, regarding hepatitis B virus, because yesterday's ceremony was a statement, right, that yes, microRNAs are very important and they have a very important role. That's why I think that probably there are perspectives to find something how to block this, because I do not have a huge information or knowledge about those microRNAs, but for the general information, it was very, very interesting, because if there are some elements who can help to HIV, then we can some kind of block it, and we can make new medicines based on this. I don't know.
It's a great suggestion. I'm sure that somebody is working in that arena now, as these microRNAs have been around for a while. But I don't, if there is a literature about them, I'm not aware of it. But if it's an opportunity for affecting the outcome of infection, that's great. I'm going to make another plug for this journal, now that you've asked about the Nobel Prize, insofar as on Pathogens and Immunity website, we have interviews with last year's Nobel laureate, Drew Weissman. So free on the website, you just go there and there's an interview with Drew Weissman. And just a couple of months ago, we had an interview, and it's on the website, with Tony Fauci, who was the director of the National Institutes of Allergy and Infectious Diseases that led the HIV research effort in the United States. So microRNAs sound good, I just can't speak knowledgeably about them.
Thank you for your presentation. It was really interesting, and I might be interested if there are some epigenetic cures for HIV.
Well, it's a good question, and a number of groups, including John Connor's case, are working on ways to affect the epigenome of HIV. There's this concept of lock and block. So ordinarily we think of viral transcription as a means for viral replication. But if you can provide an epigenetic modification of the long-terminal repeat of HIV that results in an inability to transcribe, in principle, if that is durable, one can result in a non-antiviral cure of HIV if all the cells that contain integrated provirus can be locked in position and transcriptionally silent. So yes, people are working on that.
Hello? Yeah. My question is, as a medical student from Ilyas State University, I think it's an amazing thing that you're here, and I've really enjoyed the presentation, but my question would be that, as a student, of course, how I've come to understand that there are some people who have HIV are kind of, they can be subjected, not subjected, but they can be differentiated as in how they are treated and also how they can be treated. Basically, I've read that they can be immune and they can be, wait 1 second, I'll just read it out. So basically suggesting that they can manage HIV therapy without antiviral therapy, basically. So what would you say about that?
So this is a population, thanks for asking, this is a population that we call elite controllers. And these people have viral replication that is low level, and typically with viral levels in plasma below the limits of detection by our assays, our PCR assays. So what is it that characterizes an elite controller? Very often they have dramatic antiretroviral, anti-HIV, CD8 positive T cells. Sometimes they don't. But one of the things that characterizes these people, these elite controllers, is that they are immune activated. Just like our immune non-responders, just like our untreated patients. The levels of immune activation are lower, but they are detectable. And so in some, there is a progressive CD4 T cell decline, even though levels in plasma remain below the limit of detection. These people were being studied, were being evaluated to see whether or not, what it is that distinguishes them. And often, it's a very brisk T cell response to HIV. Importantly, the neutralizing antibodies that have been isolated and expressed are generally taken from people with uncontrolled HIV. So even though they have neutralizing antibodies, they don't typically control very well.
My question was that I was very surprised that by blocking interleukin-6, you managed to show the bettering of the prognosis, so to speak. So does it imply that it's our immune system that does all the damage, basically? So does it mean that if we manage the inflammation, the prognosis should be better? Could you please elaborate on that a bit?
Well, that's a great question. And yes, the immune system in HIV is damaged. There's no question about it. Even in persons who have well-controlled viremia, it is damaged. And I want to be clear that we did not show that blockade of interleukin-6 improves the prognosis and the outcome of HIV infection. We showed that it improves the appearance of the markers that predict the outcome of HIV infection. So in order to know that blockade of IL-6 will improve health, we need to study far more people than we did in that study. That study was an initial study to provide a rationale for a larger trial. Now, here's the complexity of it. You're not going to give IL-6 blockade to somebody with untreated infection. You're going to give it to people who are on antiretroviral therapy. And both the boom, the goodness of antiretroviral therapy, this is a conundrum. And what I mean is a conundrum. If we want to study an anti-inflammatory agent like IL-6 receptor blockade, and we want to look at the effect on clinical outcomes, if the clinical outcomes in our patient populations are pretty much the same, close to the same as the healthy population, we'd have to study thousands and thousands of people to show an effect. And I'm not sure that people are prepared yet to invest in the cost of a multi-person trial. For example, when it comes to inflammation, recently a study in the ACTG showed that a statin drug, an anti-inflammatory drug, could decrease the risk of morbidity in HIV. It's a statin, like atorvastatin or pravastatin. I don't remember which statin it was, but it took 5,000 people to do that study. So it's clear that inflammation is linked to morbidity. Whether blocking IL-6 will improve morbidity, can't say. I suspect it would, but to do the study would be very difficult.
Well, first of all, a short comment to you. How yesterday's Nobel Prize on microRNA would help treat HIV, like you, I don't know. But I'm sure that today's Nobel Prize would definitely help us. If you haven't read this morning, that the Nobel Prize in Physics today was awarded to two scientists who do artificial intellect. So that's, of course, will be a cure for all the diseases, including HIV. And a short question. You said it in passing, that as we know, there are people who could not be infectable by HIV because the receptor is malfunctioning. It doesn't have an active receptor because 32 amino acids were deleted. The delta 32, base pairs, not. So the question is, and as far as I know, it's only in white population. There is no such mutation in any other races, in black, in yellow, whatever. And also it's a question which you asked, it's still sustained for so many years because it started with a race separation. So could you speculate what is the reason why we still so many people are not infected by HIV, have this mutation? It's definitely not HIV is a pressure, but something else presses it.
Vanya, I agree with you entirely. I think it's a fascinating question as to, first, let me start by saying that this mutation, this 32 base pair mutation, occurred once in human evolution as far as we know. And it's only seen among whites. Whites of European extraction too. So I think it was a mistake. But the fact that its prevalence is so high in Europeans, if it happened only once, Now it's seen the allele frequency is 15 to 20% in persons of European extraction. So how did it get to be so prevalent? I think there must have been some selection pressure that allowed a survival benefit. But we don't know what that is yet. We have no idea. It was not plague. That's not the answer. I asked Mary Carrington, you know, she's one of the bad boys of Cleveland, one of the BBC. I asked, and she's a great geneticist, I asked her, why is this allele so frequent? And she suggested that it was drift. Now, I don't think I understand drift, but I would not discard her opinion because she's very good at what she does. So is it drift? Is it selection pressure? I think it's selection pressure, but I have no idea what it was. Now let me turn your question on its end and say, when we looked at the people who had a 95% risk of acquiring HIV infection, these heavily treated hemophiliacs, of those 5% who escaped infection, they were treated with lots and lots and lots of plasma that undoubtedly contained HIV. Only 16% had this Delta 32 mutation. That means that 84% of these people who escaped HIV did not have the Delta 32 homozygous state. And we know that heterozygotes are at risk for HIV infection, plenty of those people around. We study those people, Janelle Salkowitz studied those people in my lab, and the only thing that distinguished them from the general population, you're going to love this, was that their cells were less readily activated by phytohemagglutinin and IL-2. So there was something about those individuals, CD4 T cells, that rendered them less readily activated. And as you know, an activated CD4 T cell is much more susceptible to HIV than a resting cell.